Good afternoon, everybody. My name is Abby Bronson, and I am the SVP of Research Strategy at PPMD. It's my pleasure to be able to introduce this afternoon's webinar on a novel exon skipping therapy being developed by NS Pharma. They did a webinar a little over a year ago on the program right when it was just getting started, and they're kind enough to take some time today to update us on their progress, and we're really grateful for them and for their collaboration. Before I introduce our speakers, as usual, there's some housekeeping for these webinars. We'll have time for questions at the end of their talk. So if you have a question, type it into the chat box, and we will try to answer as many as we have time for at the end. And any that we don't answer, we will get to you by email, and we'll also post those questions and answers when we post the webinar recording on our website, probably in 2, 3, 4 days, something like that. And as you know, this webinar is being recorded, so you'll be able to access it at a later date. It's now my pleasure to introduce our speakers for today, Dr. Paula Clemens and Lauren Morgenroth. Dr. Clemens is a neurologist and scientist at the University of Pittsburgh. She has particular expertise in the care of adults with neuromuscular diseases such as Duchenne and Becker. She's the medical director of Synergy, and she leads several multi-center studies focused on treatments for genetic muscle diseases such as Duchenne. So she's very qualified, and she's been in our community for many years now. Lauren Morgenroth is the COO, Chief Operating Officer for TRINS, which focuses on neuromuscular diseases. It's also the coordinating center for Synergy. She's been involved with Synergy for over 12 years and is a certified genetic counselor and an adjunct instructor in human genetics at the University of Pittsburgh School of Public Health. So now I'll turn it over to Lauren and to Dr. Clemens.
Hello, this is Paula Clemens. Thank you all for joining the call. I will start the presentation and then about halfway through hand it over to Lauren to continue it. We felt that it would be good to present at this time. This is a study that's sponsored by NS Pharma, and the first enrollment in this study began in December 2016. The study is ongoing, but we wanted to give you an update. Since it is ongoing, we won't be able to give you any specific data, but we can let you know where we are in the study. So as I mentioned, both Lauren and I are presenting this on behalf of NS Pharma, who is the sponsor of the study. Just a brief introduction to NS Pharma. This is a U.S. subsidiary of a Japanese company called Nippon Shinyaku, which is located in Kyoto, Japan. Nippon Shinyaku has collaborated with an academic center in Tokyo to develop their exon 53 skipping product, NS065 NCNP01. NS Pharma is, as I said, is the U.S. subsidiary, and they are working with a regulatory consultant firm, Pharma to run the phase 2 studies in the United States. A key player in this is the Synergy Network. That's the collaborative international neuromuscular research group, which is an academic clinical trial network focused on neuromuscular diseases and is working in collaboration with TRINS, which is a specialized contract research organization for neuromuscular diseases, and is the coordinating center for this study. I am the study chair, and as Abby mentioned, I'm also the medical director for the Synergy Network. And Lauren Morgenroth, who you'll hear from a little bit later, is the leader and chief operating officer of TRINS, the CRO that's running this study. So Nippon Shinyaku is the company that's in Japan that has developed this product. They also have factories in Japan that that produce the investigational product, as well as research laboratories. And they have offices in several places across the world, and including the U.S. subsidiary NS Pharma, which is located in New Jersey. They have a pipeline of projects in the Duchenne muscular dystrophy space. The project that we will discuss today is the Phase 2 study being run in the United States. There is also an ongoing Phase 2 study of the Exon 53 skipping product in Japan, and they previously did a Phase 1 study in Japan using this investigational product. As you can see on the slide, they have several other discovery programs for other exon skipping agents designed for Duchenne muscular dystrophy treatment. So the investigational product is an antisense oligonucleotide. It has a morpholino backbone, and it is designed to skip exon 53. This is delivered as an intravenous infusion that's given once weekly. Many of you are probably familiar with the mechanism of action of exon skipping, and I'll just review it here. In the normal situation, you have a full-length dystrophin message, which is derived from the DNA for dystrophin. And what we're showing here in this schematic is just a section of the messenger RNA which has exons 51 through 54. And if all the exons are present in the dystrophin message, then that results in a normal dystrophin protein. However, in Duchenne muscular dystrophy, there are very often deletions which remove entire exons. And what's shown in this schematic is a deletion of exon 52, and this results in what we call an out-of-frame process that truncates the production of the protein, because this particular type of disruption in the exon sequence means that the rest of the gene is not read to generate protein. So the concept of exon skipping is that one actually makes the deletion even bigger in order to bring the reading frame back into frame, and that allows for the body to produce a dystrophin that is not truncated, is missing the bit that corresponds to the deletion in the middle, so it is shortened, but it still has its two ends. And that is projected to provide a partially functional protein. So exon 53 skipping is pertinent only to specific deletions in which removing exon 53 would bring the deletion back into frame. And by looking at large databases of patient deletions, we know that this corresponds to about 8%. Those that would be amenable and treated by exon 53 skipping correspond to about 8% of patients with Duchenne. And as you're probably aware, there are other programs that are being developed to skip other exons. And if exon skipping were taken to its full extent, it could potentially provide treatments to approximately 75% of patients with Duchenne muscular dystrophy. I mentioned at the beginning that the Nippon Shinyaku had done an investigator-initiated phase 1 study in Japan. This was an open-label, escalating dose, safety, and PK study, which also looked at dystrophin to show that there was what we call target engagement. There were 10 Duchenne boys who participated in this study, and they were treated for a total of 12 weeks. This was done at a single site in Tokyo, and you see the cohort design below, where the lowest cohort enrolled 3 participants, the next also enrolled 3 participants, and then the final cohort enrolled 4 participants. And you can see that the top dose that was given in this cohort study was 20 milligrams per kilogram weekly, and that is half of the lowest dose that's used in the phase 2 trial that we are presenting to you today. So the expectation was not that there would be a significant amount of dystrophin expressed by participants in this study. It was really focused on an initial safety to make sure that one could go forward with higher doses and higher numbers of participants in future trials. But this study was completed and reported out as in Japan, and you can see that there's a press release, a link to a press release on the slide that you're welcome to check later. And then this allowed for the start of the phase 2 trials, so they were begun near the end of 2016 with the first enrollments both in Japan and in the U.S. And there have been some very important regulatory developments that significantly assist in the development of any kind of a new therapy. And these designations through the Food and Drug Administration are in recognition of the importance of the indication for the drug development as well as other features. And so they're listed here. There's what is called Fast Track designation that was achieved first, then Orphan Drug designation, followed by Rare Pediatric Disease designation. And at this point, I am going to hand the presentation over to Lauren to continue.
Thank you, Paula, and I want to also thank Abby and PPMD for hosting this webinar for us to give an opportunity to the community to give them an update. As you've heard, our first patient was enrolled in December of 2016, and this is an opportunity for us on behalf of NS Pharma to provide everyone with an update of where we stand with our program. So I will tell you a little bit more about the clinical aspects that are managed by TRIMS. And so we ended up having 6 sites in North America participate in the study. We had a site in Canada and 5 centers across the U.S. from West Coast to East Coast. I recognize this slide is a bit busy and has been previously presented, but what we wanted to do in this slide is walk you through that study design and what patients who participated went through in terms of the process. The first step in all research studies is the screening, which begins with the informed consent. That's a very important document that is reviewed with participants, their family, and the study team to be able to make sure that all assessments are properly explained. It's an opportunity to review the risks and benefits, review alternative treatments. And so that document is kept with families throughout the course of the study to be able to be used to remind them of all the assessments they have when they agree to be in a study. And so once that document is signed, patients undergo a screening process, and again, that's put in place in all projects to make sure we're including patients that it's safe for the patients to be in. So a number of assessments are done during the screening process to be able to, again, check laboratory results or physical exams to make sure there are any conditions that could make it unsafe for a patient to be in a study. Then after a 20-day period for that screening to make sure all the test results come back, patients then undergo randomization, which is a process like flipping a coin where patients will be either placed on the intervention that you've heard Dr. Clemens tell you about the mechanism of action, or on a placebo. This was a blinded 4-week period. For safety reasons, we had this blinding portion in the study. And we began enrolling patients in this 40-milligram dose. Once patients underwent their 4-week period, at their week 5 visit, they were then placed on the intervention at the 40-milligram doses. They continued on for an additional 20 weeks, at which point they reached the end of the study at week 24 and had a muscle biopsy. We did a muscle biopsy at the beginning, before the intervention, and a muscle biopsy at the end of the 24 weeks. At that point, patients had the choice and were presented with a new informed consent for an extension study and were given the choice to either consent and enroll into an extension study or choose to be followed up for safety reasons after a 30-day period. This extension study lasts 72 weeks. We did this for the 40-milligram dose. Once we made sure all patients had passed the 5 weeks and it was no safety signals were identified from an independent safety committee, we then were able to enroll an additional 8 patients with this same process with the 80 milligrams. So again, a new set of 8 patients were then either placed on 80 milligrams or placebo for a period of 4 weeks, at which point they then went on to the 80 milligrams, reached the end of the study, had their second muscle biopsy, and then were able to decide whether or not to continue into an extension study. At this point, all 16 patients have recently finished these 24 weeks, so this is why we wanted to provide these updates to the community. This just recently happened, so we have no results yet because that just occurred, and all patients have elected to continue in the extension study, like I said, that will last for 72 additional weeks. So in total, patients will be receiving the intervention of NS065-NC and PO1 for a period of 96 weeks. 16 patients in total were enrolled. Their age range was between the ages of 4 and less than 10 and needed to have one of those mutations that you heard from Dr. Clemens in that group of exon 53 skipping. Here you can see the groupings of our study assessments, and so here I'm going to actually summarize them in 2 broad groups. So we'll start with the bottom part here, and these are really the safety, and that's the primary objective of the study with safety reasons. So you can see here we did periodic blood draws, urine collection, again for safety reasons. They were more frequent in the beginning of the study and are always done at a minimum of 12-week intervals. We continue to collect every visit the vital signs to, again, make sure it is safe to undergo the infusion, but also safe as it continues on in the study. And then physical exams are, again, performed for safety reasons. The second grouping of study assessments that are being done, obviously you heard the muscle biopsies. They're being done at screening and were done at week 25. And this is, again, for the mechanism of action to be able to measure the dystrophin production. And so these are going to begin to be analyzed in the next few weeks to months. In terms of this, go ahead.
Lauren, I'm just thinking quickly before we move on that other side, because you're going down to ages for? I mean, and you didn't mention anything about steady steroids. Was that a requirement for enrollment?
Yes, thanks, Abby. Yes, so all patients did need to be on a stable dose of corticosteroids for at least a period of 3 months prior to enrolling. And so the second set of assessments that were done here were for strength and function. They're being done every 12 weeks and are, again, being done to see if over a longer period of time we see any signals of any differences. The primary objective of the study is safety, but these are being done to, again, follow along through the course of their participation. And here we're collecting some time tests, North Star, some strength measurement through the Synergy quantitative measurement system, as well as a 6-meter walk test. In terms of where we stand for the study updates, the enrollment in the 201, which is the first 24 weeks, is complete, and all patients remain active in the extension, which is 202. And up to date, we can say the intervention has been well tolerated, and we've not seen any significant serious adverse events related to the drug. And the Japanese phase 2 trial is also ongoing. In terms of timelines, both studies in the U.S. and in Japan are expected to be able to release results. In the U.S., we expect to release results for the first 24 weeks sometime around Q3. So again, as we said, it just recently completed, and we've just now starting to begin making sure all the data is clean to be able to start those analyses. And so we really want to thank the community. It's very important, the dedication of our families that are coming weekly into our sites. We've started a program called Stones of Strength, where at each visit to recognize the dedication of these families and boys are given a stone to mark the importance of these important milestones that they're accomplishing with each week of their infusions. If you do have any questions, we are happy to answer them through our trial info at nspharma.com. Both the study 201 and 202 are posted on clinicaltrials.gov, and information is also available on the nspharma.com website. So again, we really want to thank everyone for participating and for listening to our webinar. And we're happy to answer any questions that you may have, Abby, from the community or through PPMD. So thank you.
Thank you very much, Lauren and Paula. That was really informative, and you've made great progress. I have a couple questions. I think probably, this is great. In Q3, we're going to hear about some results, but considering that everything goes positively, what would be next? Like, how would you move forward? What are your plans for the future?
So at this point, the plan is to continue in the extension study. As Lauren mentioned, that extension study goes for 72 weeks beyond the initial 24 weeks of the study. NS Pharma is navigating the pathway of putting their data and proposals in front of the regulatory authorities, and that's in progress during this interval. And it really will be in discussion with the regulatory authorities and internal discussions in the company to make decisions about where to go with this in the future.
Okay, so because right now I was also going to ask about the 4 to 7-year-old age range that you're targeting. First of all, did you have a pretty good, I don't know if you can answer this question, but there's a good sort of distribution of ages among the 4 to 7 years old? Or are they more toward the older kids, or can you say anything about that?
So it's 4 to 10, or 4 to... Yeah, 4 to the day before the 10th birthday. And I think it's pretty well distributed, but I'll let Lauren address that.
Okay.
Lauren, are you on mute?
While Lauren's getting off mute, and also add to that question, I'm also wondering, there's a question that came in about if ambulant. Lauren lost a connection apparently.
Okay.
So we're wondering, was being ambulant a requirement?
Yes.
Okay.
They did have to be ambulant. That was one of the eligibility requirements.
Okay. And I asked about future plans. So, one thing that's very important to our community is the opportunity for non-ambulant individuals with Duchenne. So, I don't know if you can comment on that, but always keep in the back of your mind that's
Sure.
That's very important for everybody. So, if you can, do you have any thoughts on that?
Of course, I totally understand that, and as you know from the history of the whole Synergy group, we have really maintained a high focus on being able to think about the broader patient group. As I think most people know, when you're doing initial testing, it's really important to keep the test groups, which are necessarily small numbers, as homogeneous as possible, because otherwise it's very difficult to determine an effect of the investigational drug. Particularly, that's even more complicated in a disease like Duchenne, which has the disease progression superimposed by growth and development. I'm sure everyone on the call understands how difficult that is, and the importance of keeping the initial studies on a narrow age window, because that makes it a narrow developmental and disease progression window. But that being said, there is a very strong recognition of the importance to then take it beyond that if we find that there is safety and suggestion of efficacy in that much smaller window.
Thanks. So another question: how do you compare and contrast your program with the golodirsen program that Sarepta has? Because they're both targeting exon skipping for exon 53. So what are the comparisons?
Yes, so they're not exactly the same product, although they are both based on a morpholino backbone. So the investigational product is certainly in the same class, although as I say they were developed independently, so they are presumably different different at certain molecular levels. The study designs are quite different, and that is again a function of the Sarepta is following their development program, which in their case began with the exon 51 skipping program that resulted in a product approval. So they have had a longer history of communication with the FDA, and their program is designed by their interaction with the regulators and their own internal decisions. So there isn't any crosstalk for the most part between the two programs. So they're basically each trying to navigate the best the best path that they can to move the products, the investigation of the products forward as quickly and as safely as possible.
So they're different products targeting the same thing. Would you say that?
I would say that, yes, absolutely.
Okay.
They're both targeting deletion mutations that are amenable to exon 53 skipping that would bring the deletion back into an in-frame conformation.
Right. And another difference between the golodirsen program is the 24-week time frame that NS Pharma is using. Can you talk a little bit about why that is? Because it's shorter. It's pretty short.
Mm-hmm. So, that is really essentially a study design. These are small numbers of studies, small numbers of participants in this type of study. I would say that the NS Pharma study is a little bit earlier stage, perhaps, than the Sarepta study, which follows their phase 2. Actually, I take that back. I mean, in a way, they get to, I'm thinking of their Exon 51 program, but in a way, there's they have a history there with the Exon 51, so they're kind of further along, but they're just a little bit different in terms of the design of the Exon 53 program. Essentially the goal is pretty similar.
Mm-hmm. Yeah.
And at the end of the day, I think there are more similarities than differences.
Yeah, and I can also understand, using, our boys are precious, so use the smallest number just to make a decision and then go forward. So that's another way to think about it.
Mm-hmm.
And a question came in, will the company present the initial phase 1 data? So I think people are probably interested in what can we learn from that phase 1 data. I don't know what you can say about that.
Yeah, I was not involved in the study, so I really don't know what the publication plan is for that. So I can't really adequately address that question.
But again, it was a safety study, so there probably might not be a lot of dystrophin quantification data coming out of that. Is that correct?
Correct. It was so muscle biopsies were collected, and one wouldn't expect, based on the dosing, wouldn't expect a large amount of dystrophin to be produced, but one wants to see that from a muscle pathology perspective, it is safe, and that the investigational agent can meet its target, and so those would have been the goals of that aspect of it.
Okay. Is Lauren back online?
Yes, I am. Sorry about that. I don't know why.
That's okay. So I thought it said that your age range was 4 to 7, but I guess it was 4 to 10. Probably was my mistake. But do you have an equal distribution of patients? Is it fairly equally distributed among those ages?
So I think it's important to keep in mind there's only 16 patients, and it is a relatively narrow age range of 4 to 10, but we have patients in that entire range of 4 to less than 10.
Okay. Trying to look to see if there's any other questions. This is really interesting and great information for all of us. So, yeah, we talked about this. Do you have any plans for trials which may include non-ambulatory patients in the near future? And I think we talked about how that certainly is a desire and a wish of yours, but you have to put that in the context of a regulatory plan. Do you want to add anything to that?
No, I think that sums it up really well.
Let's see if there's any other questions. How long does it take for the infusion, and have you noticed any site reactions or irritations or anything unusual?
The infusion goes over 60 minutes, and there haven't been any infusion-associated reactions at all.
Oh, that's great. Okay. And is there ever any thought, and I know obviously this would be a completely different protocol, but with, weekly infusions, would it be possible to stretch the infusions out and maybe, I don't know how you do it, maybe give more? I don't know. This is just a hypothetical question.
Right. So, you could also ask that question of why weekly?
Yeah.
Particularly since from preclinical studies and from pharmacokinetic studies in humans, we know that the drug is cleared from the bloodstream rather quickly. So it's clearly not staying in the bloodstream for the full week. So I think there are probably a lot of questions that would need to be answered there. But you have to start from somewhere, and weekly infusions probably make sense as a starting point. I think we really, even from preclinical studies in animal models, we don't entirely understand how these agents get into muscle and get to their target. There is some fairly elegant research that's been done in that area that starts to give some insights, but we're probably have a lot to learn in that area as well, and ultimately that may significantly affect how one works on delivery, and that could be in terms of frequency of delivery and speed of delivery and amount of agent delivered. So there are many questions. I think the more you think about it, the more questions you come up with, that would ultimately need to be answered. But the first question is, can we show that the agent has an effect that's desirable, and therefore those other questions become important.
Right, I agree. Yes, that's what we want. We'll put up with a lot if it works. So, another question about the assay used for your dystrophin quantification. Was Western blot used in the previous phase 1 or only RT-PCR? And what are you doing in the phase 2? And I know that FDA, and there's been a whole regulatory process to come up with a good methodology for performing the Western blots. And did you consult with them on that? And I think people just want to make sure that is, we're not going to lose the integrity of those samples.
Certainly. So the primary outcome measure for dystrophin is Western blot. The RT-PCR looking at the message levels and also immunohistochemistry are being performed. And I think one of the things that we've been particularly pleased about in this study is just how well the muscle biopsy process has proceeded, starting with the sites and the collection at the sites, the oversight of that collection so that tissue quality is at the absolute highest level, the shipment through the central laboratory to the site that is doing the laboratory testing on the samples, and then their process. So overall, the quality of all specimens that were collected in this study was kept at an extremely high level, and therefore we feel that we're going to be able to have very high confidence in the data that we get out of that.
Okay, that's good. That sounds good. There's a question about, I think it's from the press release, that states that anemia and a slight effect on renal function have been reported as just GAEs. Is there any concern there? Are you seeing anything like that, or is it just something, you have to report everything?
Right. Hasn't come up prominently in the phase 2 study. Again, remember, we say this over and over again, but these are very small numbers of patients, even in the studies. So it is important and it is really important, and something that we focus on continuously is looking at everything that that happens in the lives of these participants, so that we don't miss an important indication of a safety concern, because when you're dealing with such small numbers, you really want to be very comprehensively looking at that, and that's really part of the program.
Mm-hmm. And Lauren, I love the Stones program. What's it called? Are you online again?
Yep, Stones of Strength.
Stones of Strength. Can you just explain that a little bit more? What exactly, do they get a stone at each visit, or?
Yeah, so this program came about from feedback from the community and from just work that others are doing, especially in cancer. They have a similar program in the cancer world, and we really wanted to recognize and thank the families for their contributions and get something back to the boys. And so what we've done here is we've teamed up with a group in Africa to empower women to end poverty in Africa. And so these women make these beads, and we have purchased the beads from them. And so at each visit, the boys get to choose a stone. And so we've steered away from the beads, even though they're beads. And then they're able to then mark how many infusions they've received through the course of their trial.
That's nice. And they like it. It's being well received.
Yeah, no, I definitely think it's a way to really, again, thank them, they get to pick the color each visit, and again, to mark the importance and dedication that they're doing in this trial. And there's 5 sites in the U.S. and 1 in Canada, is that correct? That is correct. So we have sites from West Coast to East Coast and 1 in Canada, and I'll go back to the map of the U.S. there.
Okay. And I know this is down the road, but this came up, is that have you thought at all, would this whole product be able to be infused at home, do you think, once, assuming it gets on the market and it's commercially available, or will it be something that you'll have to go to an infusion center for?
So, Paula, I'll answer that and you can fill in. So we're definitely considering the home infusion because we recognize the time away and travel time it takes to go to these just select sites that you see here. One important factor for us, because this is a safety study, is making sure all patients have been exposed to the intervention for at least a year, which would be around the week 50, 52 visit. And so if you look at the study design, we do need to make sure internally that we've passed that period to make sure there aren't any side effects that we're unaware of or that we learned. And so that is where we're using that as a criteria, and we will be working with sites to see if this can be feasible and how best we could be able to implement the home infusion. But it is something we're actively researching ourselves with NS Pharma and the medical team.
That's wonderful. That's great news. A question came in. This is probably one of the last questions that I see. Does this approach have the potential to delay the progression of disease or prevent the progression? So I think we are always wondering about that with every product.
So my initial answer would be both. Essentially, the goal of this product is to restore a partially functional dystrophin protein. So if one does that, in a way you would think about, you probably should think about it in terms of preventing progression really over the lifespan, because as we know, even though we may not see the clinical signs in babies affected with Duchenne, that the disease process is ongoing at the time of birth, and we know that because the serum creatine kinase is already very elevated at the time of birth. That's what allows us to pursue newborn screening. So I think you can think of it really both ways, but I think that as long as there is muscle tissue remaining, there is the reason to think that an intervention like this could be helpful. But keeping in mind that for an intervention such as exon skipping to be helpful, it needs to reach its target, which is skeletal muscle.
Right. So you have to wait to see if that happens. And I think there's a couple of questions in, and I know we talked a little bit about the future plans, so is an someone asked, is an NDA still planned by the end of March of 2019, and what are your plans for a phase 3? And I think when I asked about the future earlier, we kind of went over that, but maybe you just want to clarify again where we are for the next step, and then a possible NDA date.
I recognize that it's impossible to answer that question in a satisfactory way.
No.
One really needs to. It's a journey. The path to the destination is not entirely clear while you're on the journey, and the journey does involve very sort of orchestrated interactions with the regulators, which really define some of the milestones in that journey. So it's very difficult to, at this stage, when we have not even been able to do data analysis, because that has to follow after all data is collected and cleaned and analyzed, to say what exactly the path is. But we try and give you a little bit of an indication by saying that the results are anticipated to be out in the third quarter of this year. So it gives you a little bit of an idea that at that point we would start to be able to think in more concrete terms about what the next steps are.
Mm-hmm. And then you have the complicating factor of golodirsen being underway, and we've heard them talk about maybe accelerated approval. So there's it is a journey. I think that's a good way to say it, Paula. There's lots of things that happen along the way that you have to adapt and take into account as you make your plans. So if I don't see any more questions, I guess the last question, kind of a good question to end on, is right now the product is called, it's the investigational product is NS something or other, some numbers. Are you ever going to have a name for it, and do you know the timing of that?
That's an incredibly good question, and I answer.
Yeah. So I think Abby, I can probably, there will most definitely be a name. The timing is unclear. All products kind of go through this number convention, and eventually will get named. I think that's something that, like Paula said, I think it's a great question, because right now we do refer to it as NS065NCNP01. But definitely at some point it will get a name that, for all we know, will be unpronounceable like many of the other ones, but that's something that is done by other committees and other naming grouping. But at some point, likely, we will probably have a name.
Yeah, well, we'll get a generic name, or a scientific name from the FDA that will probably end in SCN, because I think it looks like they're naming all the exon skipping drugs SCN at the end. And then you'll get a brand name from your company at some point, right? I don't know. I'm just jumping to conclusions.
And that would be something determined by NS Pharma. But in terms of, you're right, the generic name is what kind of comes in between and has kind of a method, like I said, a method to the madness, because it has certain portions that are required based on the mechanism of action. So we will stay tuned on what that naming could become.
Right. Okay. All right, everybody. Thank you so much to Paula and to Lauren for their time this afternoon and their openness in answering the questions. And it sounds like this is good news, and we're all going to be waiting for quarter 3 to hear these results. And we'll post this webinar probably in 3 to 4 days on our website with a question if there's any unanswered questions. And in the meantime, you can write to me. You can write to, I think it's info at what was the email address for questions, Lauren?
Put it right back up there.
There you go. File info
at nspharma.com, and we will also be presenting some updates as well, similar to these, at the PPMD conference.
Okay, great. We'll look forward to seeing you there. Okay, everybody, thank you very much, and have a good rest of your Wednesday afternoon.